Chapter 5, Kinetoplasta, Trypanosoma and their kins. Part 2, Trypanosoma brucei. Now, this is from the section in the chapter, The Foundations of Parasitology, that is labeled Section Salivarium. Now, I do believe this has to do with the fact that these parasites are going to be associated with the salivary, the buccal opening of their insect vector. The other trypanosomes and whatnot, you're going to see that they are passed out in the feces, and then the trophozoites escape from the insect feces and penetrate into the wound in other mechanisms. With Trypanosoma brucei, the center of this lecture, we see that Trypanosoma brucei has 3 subspecies. Okay, first one, Trypanosoma brucei. Second, Trypanosoma brucei gambinzi. And third, Trypanosoma brucei rhodesians. Depending upon which of these subspecies you're discussing, as you can see here, the normal range for these that you'll be found centers primarily around the African continent. Yes, there have been some instances of brucei going up northern Africa into the Middle East, but those are few and far between compared to what you see in North Africa, Central Africa, and a little bit as you get closer to the southern tip of the African continent. The morphology and life history of brucei is a bit difficult because these organisms are, while they're single cellular, like all Trypanosoma, they are pleomorphic, meaning that between the stages there is a complete change in overall morphology. What we do find is that in the fly, okay, and it's going to, I'm a little vague here on which fly, because the tsetse fly for some subspecies, different areas that those subspecies could have a different insect vector. We see that we find the Tripomastagote Phase. Okay, the Tripomastagote Phase goes through the different regions, areas of foregut, midgut, hindgut, back to the salivary glands and whatnot. And as it's going back to the salivary glands, it's becoming the what is referred to as the metacyclic Trypamastagote. Okay, this is the infective form. The Tripomastagote that's normally associated with the gut of the tsetse fly of the insect is not infective in humans. It does not have the right morphology, does not have the right surface structures, the right protein structures to allow it to survive and escape our immune response. When you look at brucei, you'll notice that the different stages, the Tripomastagote, cyclic, metacyclic Tripomastagote, and the different stages that you find in the human, the slender Tripomastagote and whatnot, at first glance they look quite similar. Okay, elongated, unicellular organism. The flagella, as I said, folds back, associates with the plasma membrane to give it that sail look about it, to aid it as it swims through the bloodstream, works its way through the capillary beds. But on closer inspection, even as you see here on this artist rendition, if you look at the bottom left, you will see that this is where in the tsetse fly, it's a little broader, a little more rugged to it. Overall surface proteins on its surface are going to be more accustomed, more adapted, I should say, not accustomed, more adapted to survival in the gut, which is going to allow this organism to survive the digestive enzymes of the fly, digestive enzymes and whatnot that are optimal for the lysing of mammalian blood cells. In the vertebrate, the Trypamastagotes you see here, it's going to be alternate morphologies as it goes from the different slender and slender-like morphologies that are more attuned to the different regions within a human body. I'm not going to go into great detail in that. If you really want to know, please refer to this section in the chapter. What we do know is that the Trypamastagotes, Trypanosoma brucei, when it gets into the blood, that it will then spread wherever the blood goes. Wherever the blood goes, that's where you find these organisms. The lymph nodes, spleen, cerebrospinal fluid in the capillary beds, lining and nourishing the meninges. They are able to get in, they are able to penetrate through, get into the central nervous system, which is what we're going to see contributes to the overall diseases that we associate with Trypanosoma brucei. At the site of the tsetse fly bite, where the Trypamastagote is, the metacyclic, I should say, Trypamastagote is injected into the human, into the patient, you're going to see a small canker will start to form within a few days. This canker is slightly raised around the edges, semicircular. It's not a perfect circle, it's semicircular. This is where the host immune system is starting an inflammatory response due to the spread of the Trypamastagotes through the tissues and the blood and other things there. Their waste products and cellular products and the dead Trypamastagote cells. Not every one of them that gets injected into a human is going to survive. Not all of them are going to be alive when they're injected. What we have seen is that some of the people that are infected with Trypanosoma brucei will start to get large lumps on the side and the back of their neck at the base of their skull. These are the lymph nodes there are swelling up due to the inflammatory response, due to the accumulation of trypanosoma in all those lymph nodes. These swollen lumps at the base of the skull and on the neck, these are referred to as Winterbottom sign. It is a clinical symptom, clinical sign that is used in the diagnosis of Trypanosoma brucei infection. The sickness is going to be variable depending upon which of the subspecies, brucei, gambizians, that you are looking at. It's going to start off, all of them start off as flu-like symptoms that can develop more into, metabolic issues as the liver, the spleen, and whatnot become more infected. The increased numbers of the parasites starts to cause an inflammatory response, which starts to interfere with organs. If, and God willing, it doesn't happen, but it does, that the brucei does pass into the central nervous system. This is when you start to see what is referred to as the sleeping sickness. It's what the Trypanosoma brucei, all three of the subspecies, are known for, associated with, depending upon which one you are infected with. The severity of the sickness, the severity of the central nervous system involvement will be highly variable. But if it does develop to this stage, that you do have trypanosoma actively living in the central nervous system, this is when the sleeping sickness, as I said, starts to develop. Lethargy, mental acuity starts to degrade very quickly, start to see different signs of their mental awareness is starting to diminish, almost become narcoleptic. That's what it appears like. They all, fall asleep in the middle of doing things, eating, talking, walking. Almost a weird kind of dementia. If you go through and you read the textbook or you go online and do the Google of the symptoms of the sleeping sickness, you will see that, like I said, the mental acuity and all these things that are slowly starting to degrade sounds a lot like dementia. And here, the dementia that we have as an issue here in the States, they've got the same thing, but these people are suffering from it, become more aggressive as they lose the mental faculties. They become more and more aroused. They become more and more explosive in their emotional outbursts, and you'll see that they will actually tie or chain these people to trees to protect the other people in the village, also to protect them from hurting themselves. The pathogenic mechanisms, the actual how it's doing damage, what it's doing, what it's making, still relatively unclear to us. When it comes to the immunological response, it's minimal at best. Trypanosoma brucei doesn't elicit a very strong B cell response and pretty much almost no T cell response. The innate immune system, the macrophages, neutrophils, does what it can, but it is limited at removal of all of the parasites, and without the help from the innate system, it's a very tough struggle. Part of the reason why the adaptive immune system, the B cells and the T cells, have an issue with mounting a response is that brucei has the ability to alter its surface antigens. We have to keep in mind that your immune system does not learn the parasite, it does not learn the virus, it does not learn the bacteria. What it learns and mounts a response to is a part of a protein on the surface of the parasite, the virus, or the bacteria. So what trypanosomes have evolved to have is what is referred to as variable antigen types. The antigens, the proteins on the surface of the trypanosome that were inoculated, injected into the person, are not going to be seeing the same antigens a few days, a few weeks, a few months later, while still that patient is being, showing signs of trypanosoma infection. Over time, these parasites will alter how things are expressed. They will change up which genes are being expressed, which versions are being expressed. They refer to this as variant-specific surface glycoproteins. Remember, glycoproteins are the proteins embedded in the plasma membrane of living cells, and the glyco portion is the amino acids of the protein have sugar moieties that are attached to them. Okay? What trypanosome has been able to do is that it adapts. It has evolved to alter the shape of these. So about the same time, your adaptive immune system learns trypanosomes antigens, it has modified them. And we're talking a really big change, it's just a slight modification. And then your B cells your immune system and the B cells have to start over, and then start over again, and then start over again. What we now know is that it has to do with what are referred to as VSG expression sites, genes and gene portions embedded in the chromosomes, Antigen of Trypanosoma that, move. They get moved as transposable elements to the ends of the chromosomes, the telomeres. And by doing so, it gives them preferential treatment, or preferential treatment and preferential expression. So when it comes down to it, a patient with sleeping sickness, they may have trypanosoma head to toe all throughout their bloodstream and in surrounding tissues. There is a chance that the trypanosomas you could isolate, drawing blood in different areas, taking tissue samples in different areas, are going to be antigenically different. Okay, so it's not all parasites change at once at the same time to the same way, do it again 2 weeks later. No, it's this constant, like I said, every few days, couple of weeks, they will shift, they will change. Those that don't, the adaptive immune system will target them, then the innate will be able to remove them. Now, it's not to say that there's not an overall theme. There may be a certain antigenic type that is predominant for whatever reason. The immune system is having trouble learning this one. So where the others are being, bound up, opsonized by antibodies, which allows for neutrophils, mononuclear cells, and eosinophils to target them, there's going to be a few antigenic variants that B cells are just having trouble learning, thus minimal to any antibodies being made. The adaptive immune system's not quite there yet, thus the innate cells are going to target the low-hanging fruit, the ones that the antigen is already known, already learned, and is being bound by antibodies. What's interesting is that during the blood meal, when the tsetse fly takes up the Trypamastagote into its gut, we see that the variable surface glycoproteins, the VSGs, will be replaced by a protein known as procyclin. The procyclin seems to give the Trypamastagote in the tsetse fly gut some protection from the digestive environment. As the parasite goes through its different stages in the tsetse fly gastrointestinal system, procyclin's there until it starts to traffic back to the salivary glands, the buccal cavity, where the procyclin goes away. It's, recycled back into the cell, and the VSG, the variable, glycoproteins, the surface glycoproteins, will be re-expressed so that when the metacyclic goes back into the next host, it has some protection against the immune response. When it comes to diagnosis and treatment of brucei and its different subspecies, one of the big ones is a blood smear. Look for the visualization of the parasite in the blood. There are other tests because the visualization just tells you a trypanosome. It does not tell you exactly which species. There are other tests like the CAT test mentioned in the textbook that allow for a more precise, definitive diagnosis for genus, species, subspecies, so that if there is any drug interaction that could be a concern, they can target the drug to the specific subspecies. There are drugs that can be used. Arsenic-based drugs seem to be preferential right now. Problem is, while they work great against trypanosoma, have a high level of side effects, which includes liver damage, kidney damage, some central nervous system damage. So you're trying to fight off the sleeping, keep it from, progressing to the sleeping sickness, but you're already doing your own damage to the brain. Something needs to be taken into account. If caught early enough, before there's any central nervous system involvement or whatnot, they have been having some good indications with the drug known as Suramin. Okay, this helps resolve in the early stages so it doesn't progress and you do not end up with, tissue damage, organ damage, or central nervous system damage. Once it has progressed, if it's allowed to progress to invasion into the central nervous system, prognosis is poor. Basically, it's going to be supportive therapy, help the patient. Yes, there can be some drugs, the arsenic-based drugs can be used to remove, but the problem is once the tissue damage is done to the spinal cord, to the brain, there's no coming back from that. When it comes to epidemiology control, basically it's go after the vector. Remember, trypanosomas are either in the human or they're in the tsetse fly. So if you want to control brucei spread, you go after the vector. Spray, they were at one time using DDT and many other sprays trying to get rid of, trying to cut down. There are other avenues that they're trying to look at now, using mosquito nets, things like that. But the problem is tsetse flies are daytime, so you're going to be out, you're going to be active, and that's when they're going to come in for the bite. So they're looking at other options there.